I'm really excited to be here talking to you about what I do. I think it's very important for us scientists to come together and talk to the general public about why we squirrel away in our labs for so long. But I'm also very nervous. I am very, very, very nervous. You see, I have quite a lot of social anxiety. It's the type of anxiety that, for whatever reason, makes the idea of initiating contact with people kind of a stressful situation. Incentive and motivation drives us all to interact with rewarding stimulus. It's crucial for human survival, and it's ingrained in our basic brain functions. Similarly, staying away from bad or hurtful or harmful stimuli or situations is also similarly crucial for our mental and physical well-being. This mix of signals in our brain that direct us toward positive things, but also steer us away from negative things, is ultimately what directs our decision-making and our motivation. Now, social interactions are themselves intrinsically rewarding, or at least they should be. Individuals with robust social networks tend to live longer, healthier. So why do people with social anxieties face such struggles to engage in a pretty standard behavior that's ultimately just rewarding for us? Are there some signals in our brain that tell us, no, don't do this, this is bad, when you're, just hanging out with a friend? Is there a way that we could take those kind of aversive signals and just quiet them down so that people can live their lives a little bit better without stress or fear or doubt? Well, there's a number of other psychological conditions that are associated with an alteration or lack of reward-seeking behavior, including major depressive disorder and drug abuse disorders. And a molecule named dopamine may be a central player in why we experience these things. I didn't even introduce myself, although I guess the screen did. I'm Cameron Oram. I'm a PhD student in neuroscience at McGill University, and I work for a lab that's interested in understanding the types of information that is conveyed by the molecule dopamine. Dopamine is naturally secreted throughout your brain, and its release increases when you encounter unexpected, rewarding stimulus. Experiencing enough dopamine is enough to motivate future behaviors to engage with that similar rewarding experiences. And sometimes dopamine is all it takes. In the lab, if you take a mouse and you put it in this chamber with two nearly identical rooms, and you stimulate dopamine secretion only when the mouse is in that first room, in future trials, that mouse will go out of its way to spend as much time as it can in that first room, the room it associates with dopamine and some sort of rewarding experience, even though there's nothing really intrinsically rewarding about it. Now, if you experience something aversive, like let's say you drink a glass of milk, but it's like sour milk, something else happens in your brain. Most dopamine neurons actually end up decreasing the amount of dopamine that you secrete. But a subset of neurons will actually increase the amount of dopamine that they secrete, similar to what you would see in like a reward-responsive neuron. We could categorize those dopamine neurons that increase secretion in an aversive stimuli as an aversive responsive neuron. A mix or balance of reward and aversion that is encoded by dopamine means that someone with anxiety might be experiencing too much of an aversive dopamine response and not enough of a rewarding response. It also suggests that you could possibly quench or inhibit those aversive responses that would encourage people with anxiety to engage in social contact and social interaction. But a crucial step before we can even test this hypothesis is to identify which of these cells are actually reward responsive versus aversion responsive. One of the reasons why there are different dopamine responses to one stimuli could be explained by the inherent diversity and heterogeneity of the dopamine neuron population. Dopamine neurons make up a relatively small proportion of total neurons within your brain, less than 1% of them. But within that small pool of dopamine neurons, there exists considerable cellular heterogeneity. Now, all cells can be categorized and classified based on the different combinations of genes that they actively express as these blueprints of information that we call transcripts. These blueprints are essentially your genes being put to use in a cell. And the difference level of blueprints within one cell is why a neuron is not a heart cell, and a heart cell is not a skin cell, even though they share the exact same genetic information. A technique called single-cell transcriptomics is used frequently in the lab to actually quantify and compare the level of those transcripts, of those blueprints, from cell to cell, so that we can assign an identity to an individual cell as either being a neuron, a heart cell, or a skin cell. Considerable work has already been done in identifying dopaminergic or dopamine cell heterogeneity, with at least 6 different subtypes of dopamine neurons existing within mice. This suggests that there's definitely potential that one of those subtypes is the type that responds positively to reward, and one of the subtypes responds positively to aversion. However, consensus is mixed on the identity and the relative number of the different subtypes, due in part to technical constraints. Now, when you're doing traditional transcriptomic experiments, you take a brain tissue and you mush it up into a single cell suspension. Then you record all of the transcripts and blueprints that differ from cell to cell. But doing so means that you lose a lot of information on the nature of the cells that you're analyzing. Specifically, you lose information on where those cells come from in the brain. One project that I've been working on in the lab is to utilize a technique called spatial transcriptomics to characterize the dopamine neuron diversity within mice. In an experiment that I've conducted, we're able to take a brain tissue and run spatial transcriptomics on it to identify all the different cell types that exist within the brain. And so far, on just one section, we're able to identify at least 30 different types of neurons and the supportive cell structure called glia. You can observe and see, if you look at the top of this brain region, in a region called the cortex, this nice rainbow layering of different cell types. By the way, the different colors represent a different cell type. We were also able, in this experiment, to identify the dopamine neurons, which are listed in the image on your right. Now, as we collect more data and we get a larger value of these cells that we're analyzing, we'll be able to take that dopamine population and apply our own color coordination system to it that indicates these are all different cell subtypes. And most importantly, we'll be able to put a label onto each of these individual cell types, some sort of identifying information that says this cell subtype is defined by the expression of these genes. But that label is extremely important for the next component of my research. When you do transcriptomic analysis like this, it's sort of like going to a family reunion and taking a photograph of everyone standing around. You get high resolution, you get a lot of information about what people look like, what they dress like, maybe if they're getting too drunk that day. But you don't get to know a lot about the actual characteristics or personality of the people in the photo, right? It's the same thing for looking at cell diversity. We don't actually know anything about the function of these cells, whether they respond to reward or aversion. So in order to figure out the actual function of these different cell subtypes, our lab is utilizing a technique called fiber photometry in combination with another technique called intersectional genetics. Let's say we start out with a diverse subtype population of these neurons here, A, B, C, D. And we want to look at the function of a single neural type, let's say neuron A, We can use intersectional genetics to artificially express a protein just in neuron group A that glows green whenever that neuron is active. At the same time, we can take a different protein that glows red whenever the neuron is active and put it into the rest of the population of dopamine neurons right there. This allows us to label a specific subtype with something that tells us when this neuron is active. When we collect the light, we can separate the two populations' neural activity on a graph like this while an animal is engaging in social rewarding or even social aversive stimuli. This will allow us to identify whether or not these subtypes sort of function differently in just natural behavior. This project is still very much in its infancy stages, but I hope you've come to appreciate that these two techniques, when used together, have really, really powerful potential when it comes to studying a complex brain and the complex functions associated with it. The use of spatial transcriptomics to identify single-cell heterogeneity within the brain, in combination with intersectional genetics that will allow us to actually assign a specific function to that single cell type, could definitely be used for targeted therapeutics or modulation therapies where we specifically alter the activity of one single cell type to see what happens in a number of psychiatric conditions, including anxiety. I hope one day to identify my reward-responsive neurons and my aversion-responsive neurons so that we can find a better way to limit the fear, the stress, the anxiety associated with life, and to bring balance back to an imbalanced brain system. Thank you so much for your time.